Good morning, everyone. My name is Filippo Genovese, and I'm the president of the European Federation. So welcome everyone to the first webinar of the series of awareness. Please let me spend a few words about the Federation, and we are a quick presentation I'd like to share with you. So you should be able to see my screen now. So this is the last picture was taken during the last General Assembly, which took place in June in Barcelona. So I'm pretty sure you know that CMT doesn't stand for Country Music Television. It's the most frequent inherited peripheral neuropathy. We'll hear more about it from our today's speaker. There are a lot of issues with CMT. So what happened is that in 2016, a few of us, a few patient advocacy group members and delegates meet up in Venice, and we felt that was something we could do better and together. So we founded the European Federation. As a federation, we can gain more power and legitimacy to advocate with national European health authorities. We can attract funds and try to inform the public and patients for awareness and catalyze new patient advocacy group formation in countries where there is no group at the moment, exploiting the experience and expertise and know-how of members who are more navigated in that sense. So we started with that. We were six, now we are 12 full members and 10 associate members, covering more or less all the patient advocacy groups across Europe. You can browse our website when you can get a lot of information. And one of the main focuses of the Federation is raising awareness for CMT. So we run many campaigns during the past year, focusing on different audiences, patients, doctors, and they were pretty effective so far. And this is the last theme, the last awareness campaign, which actually is currently ongoing. And this year we focused on real symptoms and showing kind of invisible symptoms and that are related to CMT, and sometimes they are not stressed enough, in our opinion. We have a dedicated website which is called cmt-awareness.com, where you can find a lot of resources and patients' testimonies and also other webinars. Main highlight of the last year was the Paris first CMT Specialist Conference, which saw a lot of specialists from all over the world getting together and working on for a better future for CMT patients. So a result of that conference was the foundation of the Europäische CMT Forschungsgesellschaft. Sorry for the pronunciation, German, for the German attendance. It's actually an association of CMT researchers which are working together and attracting funds side by side with patient organization. Another activity of the Federation is lobbying at an European level. So we prepared last year an election manifesto for the European election. These are the board members. And we are struggling, we are working to boost research for CMT, supporting researcher, providing support to laboratories and pharma companies, and again, sensibilize the patients and their doctors about the existence of the drug. So our future plans will be briefly a second CMT specialist conference, which will take place next autumn, next fall. We are organizing dedicated web meetups on specific topics and training for members. And we are trying to contact our patient advocacy groups from all over the world to expand our network. So I kindly thank you for your attention. This is the very first picture which was made during the first awareness campaign. And please save the dates, because there will be 4 more webinars coming. We'll keep you posted in our social media, and we'll let you know how to participate to these events too. So thank you for your attention, and now it's my pleasure to introduce you to our today's speaker. So the title of the webinar will be Seriousness and Early Symptoms of CMT Usual Manner. And the speaker today is Dr. Maike Dohrn. She's an award expert in the field, and she has a lot of experience with genetics, ultrasound, and please, Maike, the stage is yours.
Thank you very much. Let me share my screen. So you should be able to see my presentation. Let me, okay, is it in presentation mode now? You can see it? Okay, and you hear me well? I'm close enough to the, great, okay. Thank you very much. Thank you for the kind introduction, and thank you so much for inviting me here. I feel honored to be able to present some of my experience, and I'm really, really happy to also spark some discussion, because I think webinars are great to not only teach like teachers do in school, but also to actually really discuss and get new ideas. I have a presentation that is intended to last like 30 to 45 minutes, depending on how much we want to dwell on specific topics. And then we have, I think, enough time to actually, have a little Q&A and discuss. So this is supposed to be relatively casual, so that everyone feels happy to actually intervene and ask whenever you don't understand anything, or you have a specific question. And I was asked to give a little bit of a user manual. I mean, of course, CMT is not a device that you can explain step by step, and it's such a diverse disease or group of diseases that it's also not easy to explain a how-to CMT. But I think at least what I try to bring across is what can we do, what do we have to think of, and what are some perspectives. Let me see how we can start. Disclosures. I have in the past worked with several pharma companies, which I think makes sense in my research topic. And I have also received some substantial funding, which I'm really thankful for. I right now work in Aachen, in Germany, at the Neuromuscular Center, where I've also been trained during my residency. And I'm now leading the group, the research group of hereditary neuropathies there. And I had like very much of my training has also taken place in Miami at the Hassman Institute for Human Genetics in Stefan Züchner's lab. So a lot of these things that come into the presentation now are partially from Miami and partially from Aachen, and also other centers such as Porto or Tübingen. This is in many of my presentations my first slide, so just to get an impression, and you will see many patient pictures actually during the talk, and they are all from patients that I have met, that I have worked with throughout the years. Most of them came to my outpatient clinic. In some cases, I came to their place, or clinicians who saw them more often and provided me the pictures. So I am allowed to show these, and other sources that I used will be referred to where they come from. Okay, so first, just a little overview on the phenotype spectrum. We will go into details specifically on CMT on one of the following slides. So my research is focused on all types of hereditary neuropathies and axonopathies, because we know that axons share some mechanisms, and even though many years ago experienced neurologists defined several disease entities such as motor neuron diseases or small fiber neuropathies, or everything that is in the middle of the spectrum, and this is CMT in the European nomenclature, we know that nerves and mechanisms and genes and diseases do not fully respect our classifications. So we have to think a little bit outside of the box. And I think this is an advantage if we learn and if we collaborate and if we are not 100% purely focused on CMT, but look to the side and learn from other diseases that maybe already have treatment options. So what I work with is the whole spectrum of hereditary neuropathies. And of course, CMT is one of the most common entities that I have in my outpatient clinic. And we try to organize the way how we approach these diseases from a functional level. So are the symptoms mainly sensory, or are they mainly motor, or are they somewhere in between of the spectrum? And then we have, oh, now it's working, we have the genotype spectrum. And if you look carefully at this slide, you might notice that actually I cut down here. So a part of it is missing. So this is not even everything on CMT. There are more than 100 genes that have been associated with CMT so far. And we assume that specifically for the exonal entities, we only know about like maybe 50% of them. So it's quite possible that this list is really incomplete to date. So this is everything I want to say here, because basically you do not need to know all the genes by heart. You do not need to know all the 2Bs or 2A1, 2A2 variants here, because this is something everyone can easily look up. But what is important is to know that there are so many different genes feeding into the same disease entity, and that with every gene, there's a different gene function, a different protein, a different pathway, different mechanism. And today we quite don't know really, at least our understanding is incomplete, why so many genes and so many different mechanisms on a cellular and molecular level can actually cause so similar symptoms. And this is on the one hand a little bit disappointing or discouraging, but on the other hand, this is a great challenge for us, because somehow nature is complex and needs a lot. Like apparently it seems like if only one part of a big picture is missing, this can already cause disease. Yeah, let's learn from it. This is a great overview from a quite recent review that I would definitely recommend to look at for all the different treatment approaches organized by genes, and here organized by cell types. You know that there's axonal neuropathy and there's demyelinating neuropathy based on the cell types that is mainly affected. And here the authors quite beautifully organized the genes and then different ideas of how this mechanism could be targeted. And those three are the ones that have already reached the stage of clinical trials so far. So for now, I think, again, nobody needs to learn this by heart. I think a very important take-home message at this point is that there's hope, right? That there are many ideas, and ideas are the beginning, right? The beginning of a story. And to understand what ideas might make sense in the future, we need to know a lot about the cells and cell types, how they function in healthy and disease stages, and also about all the different genes that are involved. So this is an overview that I like to show how do we actually reach what we want to reach, right? I mean, whoever is present today, we all have the same goal. I think this is important to state right at the beginning of the webinar. We all have the same goal, and the same goal is this one: treatment, right? To treat, not, I mean, ideally, I could even say cure, but I'm a bit careful with this word because cure is big, like that's a big deal. I would already be quite happy with treatment, like slowing down the progression, or even stopping progression. That would already be a milestone. So let's define this as our ultimate and common goal, and the important step to get there is clinical trials. And what do we need to get there? From my point of view, I think the very first step, and I will illustrate this later, is to find the genetic cause of the disease. Like genetic sequencing and data sharing is essential to get there, like to understand the causes, and then also to do functional studies, which means to understand what these genetic defects, what is their consequence? Like, why do cells decompensate? Why can't they just keep working, right? This is important for us. And for that, we need a lot of wet lab work. And here you see my lab bench in the background. I'm right now currently not in the lab. I'm working from home these days, but this is always my background. But you see my original lab bench here, and we need animal models and we need computer simulations. So a lot can be done here at this point, but that's not enough. We need more. We need to understand phenotypes. So we need observational studies. How are these disease entities in nature, in their natural environment? So what is the goal? Like, what actually do we want to slow down? We need to do genetic studies to examine other family members. We need to work on human tissue from histology, like from biopsies. And we need to share all this with other centers and registries. And also later, ideally, when a drug has been approved, in open label extension studies, so long-term observations of a drug and their effects, to better understand what are the benefits, what are the long-term side effects, for instance, and also to which age groups, for instance, can we recommend those. So this is quite complex, and nobody can do this alone, right? So what I do is I work on genetic sequencing. I have done some wet lab, but that's not my main focus. And I do a lot of natural history observation studies and working on clinical trials, but always in a big network. And I think this is essential to demonstrate. And this is also what ECRA is about, what like all CMT associations are about, to join our expertise and to work together, to collaborate as a network. Not just clinicians, not just researchers, not just patients, but not just companies, like everyone together. Okay, one example that illustrates how fast this can actually go is a CMT sort. Many of you might have heard about it. Andrea Cortese found this gene together with Adriana Ribello and other colleagues of mine. And they wrote a great paper published in Nature Genetics back in 2020. And this is most likely the most common axonal recessive CMT subtype, because the most frequent allele shown here and illustrated here in the Sanger sequencing frame shift mutation, you can really see how the frame is shifted. This allele is so frequent in healthy controls in heterozygous state, so just one allele. And since it is so frequent, relatively frequent, just by likelihood, it will happen every once in a while that two carriers marry, have kids, and then suddenly have affected children, right? So this is the recessive mode of inheritance, and this seems to be quite a common disease. And the mechanism is what makes it so special. So the gene sort that is mutated here causes a loss of function in the enzyme sorbitol dehydrogenase. Nobody needs to learn this by heart at this point, right? There will not be an exam at the end of this webinar, but I think it's nice to understand how all this biochemistry and genetics and patient work actually emerge and come together. And here, if this enzyme is not working, sorbitol, that is derived from glucose, cannot turn into fructose. So sorbitol levels increase. And if this happens in nerve cells, in axons, in Schwann cells, in nerve compartment, sorbitol levels go up in the nerves. And the idea is of this treatment option that has already reached the stage of clinical trials is that a small molecule, an aldose reductase inhibitor that patients take orally, will inhibit this upstream enzyme here, so that sorbitol is not even being created in such amounts. So sorbitol levels can be reduced. And interestingly, the first discovery of this gene was in May 2020, and only 2 years after that, this phase 3 clinical trial has started. So this is, I'm not going to dwell on this much longer because there will be a separate webinar on this, but I think what is really remarkable is that all these steps that I showed here, right, remember, have been taken in just 2 years. And that is, in my opinion, absolutely crazy and only possible if there are structures and networks that have already been working together before. So this needs to be prepared. You cannot start from scratch with every new mechanism. We need to be ready for trials. And this is one of the aims of these research networks that Filippo has just introduced. So today we will talk about CMT. What is CMT? I think I don't need to introduce that in very much detail, but I think it's important to see that we're all on the same page and everyone talks about the same thing. And then I will illustrate throughout the webinar some common and some rare forms. I will try to show some typical symptoms. What do we know about the course of the disease, the natural course? Then what are important steps in making a diagnosis? What are consequences of a diagnosis or even early diagnosis? And then really important question that I get asked a lot is what to do if the genetic test is negative? And then also, when should other family members or even children also be tested? So you have already seen these 3 guys at the beginning of the webinar, Mr. Charcot, Mr. Marie, and Mr. Tooth. These pictures come from Wikipedia. They together have defined this disease. But I think what is more important to know is how this actually looks. Most of the patients develop their first symptoms in the first decade, so before the age of 10 in general. But then there are also later onset forms that begin in a teen age or even later in life. So this doesn't exclude that there can be later onset forms. Very typical for clinicians is that the Achilles tendon reflexes are lost, which is not something patients will notice themselves, but what becomes very obvious at the first doctor's appointment. But what is obvious is foot drop, like difficulties running or even walking, and then a loss of muscles, atrophies, especially in feet and calves, and then pes cavus, which is seen here, and I will show further pictures that the feet look basically hollow because the muscle is going away, and then also the skeletal architecture of the foot changes because the remaining muscles are not in an equilibrium anymore. So usually the pattern is most distal, which means far away from the body trunk, and perineal, which means it is pronounced in the foot elevators. And then we have weakness and atrophies. We have this typical steppage gait. Sensory means is typical that there are more minus than plus symptoms, which means that there is more sensory loss other than neuropathic pain or tingling, which can also occur, but it's not that predominant and is very dominant in other types of neuropathies. So this is quite good to see if something is rather CMT or diabetic neuropathy, for instance. And then we have deformities. Pes cavus, I've already shown that, claw toes, also sometimes scoliosis and others. And then there are possible signs that might be specific for some genes or gene clusters, like thickening of peripheral nerves, so that you can maybe even palpate them with your fingers. Hearing loss can occur, optic atrophy can occur, macrocephaly can occur, but only associated with specific mutations in specific genes, so not in general in all CMT subtypes. Those are some clinical pictures. This is a female patient in her early 50s. You can see that her calves are really thin and that she has foot deformities here. Those are her hands, also muscle loss. She has an axonal CMT subtype, and we have not found the genetic cause yet in this case. And here we see mother and daughter. This is the mother, and this is the daughter, and this is from the other perspective, mother and daughter. Again, they both have a PMP22 duplication, so classical CMT1A, and they're both affected. And what is interesting, I think, here to illustrate is that the mother had several foot surgeries back then, and the daughter has not. So foot surgeries is also something that is like very much in the focus of discussion. Is this actually useful or not? Some patients say yes, others say no. Some have good experiences, some have bad. There's not one typical procedure that applies for all types of feet. So it's very difficult to standardize those results. And back then, when this now 50-year-old woman was younger, it was really common to do foot surgeries. And now it's not so common anymore in Europe. So it's interesting to see. And it would be great to have more standardized knowledge on how this is actually working and what patients to recommend it to, but there's not much data, unfortunately. So here we have videos. This is one patient in my outpatient clinic. He's a male, he's in his early 40s. You can see that he has problems elevating his feet, and he can also not stand on his heels or tiptoes. His gait is a little bit unsteady, and specifically you see that his calves are quite thin. He's, other than that, really sporty. He's always going to work by bike, but he's wearing specific special shoes in order to stabilize his ankles, and he has short neuropathy. And then we have another patient, same age, early 40s. She is wheelchair bound. She's very severely affected by CMT. I have more pictures here of her. You can see that also her hands are severely involved, loss of muscles here and here, and also even her thighs are affected, which is the reason why she's why she's wheelchair bound as well. And she carries an ITPR3 mutation. So this is a demyelinating type of autosomal dominant CMT. So what do we know about the course of disease? I have two examples here illustrating some data on the natural cause. This has been published by Isaac Shu back in 2023. And this is CMT. This is a huge data set from the INC, Inherited Neuropathy Consortium, that has been collected through many, many years, actually prospectively. So these are really pure and valuable data. For this, we analyzed more than 20,000 patient visits. So this was a lot of work. Shout out to Isaac. And here we can see that this is progressive disease. This is the CMTES score, a score that combines many different clinical exams and also some questions that we ask the patients in order to quantify disease severity. And we can see that there is a linear correlation with age. So this is a progressive disease correlating with age. And what we wanted to know, I think this is the even more important question, is does the velocity of progression change over time? And actually, no. Here we see the change in the score over the years, here correlating with age, and it's completely stable. So even though some patients experience a sudden worsening, which can definitely be true for each individual, overall, looking at it from a statistical point of view and at the whole cohort that we have analyzed, progression and velocity stay stable over lifetime. So that means if you are one of the lucky ones who had a very slow progression all your life, that is good news. That means that it will not suddenly, most likely, will not suddenly progress much faster than before. But on the other hand, that can be bad news too, of course, if you are not one of the lucky ones. So this applies for CMT1A, the most common type of CMT, which we have many data sets for, but for others, we do not have so many data sets. The second example that I brought here is CMT sort, and this is data from our global natural history study that I have organized together with Stefan Züchner and Andrea Cortese. The paper is currently under review in Brain, so we are really much looking forward to hopefully seeing this published very soon. And these are some of the data that we have analyzed and shown in several presentations. So in this study, we had access to 185 patients, but full data sets were only available for 145. So compared to those 20,000 visits, it's not much, right? I mean, I think this is important to say, it's not much, but it's also a very young disease entity, at least to our knowledge. So we will learn a lot more from this. But this is also quite a late onset, actually, right? In teenage, most of the patients are 14 to 16 when they notice the first symptoms, so a bit later than CMT1A, but also relatively slowly progressive. So in our cohort, we only had 2 patients who used a wheelchair, and one of them was relatively old here. So this is not a statistically solid information with 2 patients, but it means that this is a rather slow disease. And here we looked at, we again correlated with age, and we looked at foot dorsiflexion strength. And there are 5 different, in total 6 levels, how we can classify this strength. And here we also see that it takes a lot of time, like about a decade, to lose 1 point. So that's again showing that this is a slowly progressive disease. So we figured that it's about like 3% reduction of muscle strength, distal muscle strength per year. So rather slowly progressive, which is important for us as researchers and clinicians to know, because if we want to see clinical improvement through a medication, we need quite a long observation time in order to be able to make a difference, right? So this is important to know. For the more rare entities, we do not have much data. So it's important to gather those data for sure. So what I've shown you is the most common dominant and demyelinating disease and the most common recessive and axonal disease. So 2 quite good examples, but there are of course more CMT subtypes, and we still need to collect those data. And again, here a little remark that we need registries for this, and we need clinicians to participate, and we need patients to participate in order to get that knowledge. So how is CMT even being diagnosed? And again, we see a patient, we see some videos here. This is part of the clinical examination. Many of you might know that we can do quite substantial sensitive testing and ask patients whether or not they notice some vibration, and ask them to say stop when they do not notice it anymore. So this is a good test for clinicians to perform, to get some knowledge about the clinical or sensory status. And of course, we also look at the patient's gait here. So here again, we see steppage gait, and the patient is trying, later in the video, will be trying to walk on heels and tiptoes. So we look at patients, we give them some tests to do. We also check on reflexes. We assess muscle strength, as I have mentioned before. So a lot of clinical tests that we do, and this is absolutely crucial. We sometimes forget about it in the age of fancy MRI scans and new ultrasound methods and really cool genetics methods. But still to date, and I will come to this at the end of the talk again, CMT is a clinical diagnosis. So if you think you have CMT, go to a neurologist or pediatrician that is specialized in neurology. And this is a clinical diagnosis. A genetic test cannot exclude CMT because not all genes are known to date, right? So if you have CMT or not, it does not depend on a genetic result. The genetic test has the purpose to define the underlying genetic cause of CMT, right? But if you have it or not is a clinical diagnosis. And then something that patients really don't like, and I understand why, is nerve conduction studies. So I think I don't need to explain this from scratch. Many of you know it. neurologists, we apply some electric stimuli at defined, by anatomy, defined nerve points, and we measure how long it takes until a signal travels across a nerve and until an action potential is being developed. And this is actually a really, really good method to understand how the nerves function. So everything else, like ultrasound, MRI, biopsies, great stuff, right? I'm gonna come to that too. But this only shows how the nerves look. And this test is the only test still to date that can show how the nerves work, how they function. So we can actually find them, I think it's an Italian word, in flagranti, like we can really see how they do or how they don't do their work, right? So still to date, even though this is a very inconvenient test, it is absolutely crucial for us to understand what's going wrong. So if your doctor asks you, please, can we do another nerve conduction test? You can ask why, but sometimes it's really meaningful. So here, for instance, we see that the nerve conduction velocity of a section of a nerve is only 15, and it is meant to be above 43. So this is clearly a demyelinating CMT subtype, and you can also see it because these action potentials are really scattered and long. And here in the lower curves, we can see that the action potentials even leave the screen. So we would have to zoom out in order to capture the full potential, which is not normal, right? So seeing this in clinic, I already know, oh, I got to test the PMP22 duplication. It's a really good indicator. And there are other demyelinating forms, of course, but this is the most frequent one. And actually, this is a 22-year-old female with CMT1A. And then we have nerve ultrasound, something that I personally like a lot. It is not painful. It is not invasive. You can easily repeat it. It's a lot of fun because you can see your nerves on screen. Patients can follow along. Patients like it much more than nerve conduction studies, but we don't see these two methods as alternatives, but as complementary. So this is my dear colleague Nathalie Winter from Tübingen, Germany. I think she's one of the best nerve ultrasound experts alive. Like, personally, I believe that. She's really, really great and a good friend of mine, and she taught me so much. So we are doing some studies on muscle and nerve ultrasound in order to assess if this would be a potential biomarker for clinical trials. And what you can see here is an incredibly enlarged nerve in CMT1A. So we cannot differentiate the myelin and the axons yet. The method is not good enough for that, but we can see that the nerve is really thick. And what we explain to patients is that you can imagine if you have a cable and the insulation layer that is wrapped around the cable is damaged, for example, because your rabbit bit in it or whatever, like it's damaged, then you might put some tape around it in order to at least make it a little better, right? The conduction. And this is exactly what the nerves do as well. They wrap something around it in order to compensate for the demyelination. And this is what we can see here, that the nerves are enlarged. And this is a cervical root, like coming out of the spine and then spreading out to become arm nerves. And here we see that this root is also very much enlarged in CMT1A. So these are pictures, again, from CMT1A, which I decided to show because this is an analogy to the nerve conduction studies that I have shown in the earlier slide. What we also like to do in Aachen, mostly on a research basis, is quantitative sensory testing. Most of you might not know this method because it's not so common all across the world. It's quite common in Germany. I personally like it a lot because, again, it is non-invasive. So what we do is we can test several sensory entities like warmth and heat, putting this probe on the skin here and slowly warming up the probe or cooling it down not in a dangerous range of temperatures for sure. So this is nothing too painful or too dangerous, but and patients have a button to press whenever they notice some change in temperature. So this is a method that we repeat a couple of times, and then we can quite well quantify the sensory thresholds for several sensations. And this is for light touch, so very fine filaments here applied to the skin, and patients tell us from which strength on they can notice it. And this is for pinprick. So there's a difference in just noticing a stimulus and noticing that it is sharp, that it is like almost a tiny little bit painful. And so we want to know from which threshold on patients notice the difference between a sharp and a normal stimulus. And now genetics, right? This is one of my favorite things to do, but I like all of them. So I'm going to show you 3 different types of methods. Again, there's not going to be an exam. You don't need to learn this by heart. It's just to give some insights. So the most traditional and oldest method is Sanger sequencing. So this is a single gene with defined primers. So a specific region of one gene. So this is a great method and still considered the gold standard if you assume that there is a specific genetic defect. So great, for instance, to test relatives, other family members, because you already know what the genetic exchange of interest is, and you can just look for that specific mutation by Sanger sequencing. And this is how the results look. And here again, you see the most common frameshift mutation in the SORT gene. And usually this should be like all aligned. And here you see that there are 2 peaks, and that means that there is something that is not in line, right? So it's a quite good method to find such type of mutations. This is a next generation sequencing data set. The most common one is exome sequencing, and here we can also see that this is in an exon region. Exons are the parts of the genome that are coding, so that really translate into proteins in the cells, and they are about like 2% of the whole genome. And here, by next generation sequencing, we can sequence many genes at the same time, and the method generates several reads from both directions, like forward and backward, or reverse. And so with that, we need a bioinformatic pipeline to align the data. And then we can see that here in this example, there is a T that doesn't belong there, and it occurs in about 50% of the reads. So that means that it is heterozygous. So this is also how we look at data sets. And this is an example from a whole genome that we sequence. So everything, a lot more data again. And this is important to also capture intronic variants. And this is an example published by Andrea Cortez again in the RFC1 gene, where there is a repeat expansion. You can see here that this is the control, right? And here is something extra that doesn't belong there. So this part is expanded in the patient. And since it is in an intronic region, which is not coding, so not translating into proteins, but is rather regulatory in the genome, that would not have been found by classic exome sequencing. So whole exome sequencing still contains some holes, right? This is important to know, even though the method is becoming much better. And then just very short, I like to mention biopsies. Some patients might have had a sore nerve biopsy in the past, and this is still a really valid and good method to look at nerves. Here you can see many different nerve fascicles, and you can see here that some of the myelin around the axons is really thick and enlarged, which we have already seen in the ultrasound earlier, right? So this is also an analogy here. And this is electron microscopy, so at a very high nanometer level, looking at those nerves. And you can see that there's too much tape wrapped around the nerve here. as an attempt of compensation here. And then there are many different exons that are not really properly myelinated. So they are sick, they are not working correctly. This is how it should normally be right here on this side. So this is a great picture. The problem with nerve biopsies is that they are invasive, right? Part of your nerve needs to be cut out. And in many, many cases, this leaves not only a scar in the skin, but also a scar in the nerve. So patients experience some electrifying sensations or tingling in that area. And also part of the foot stays numb in that area. So this is quite an invasive procedure. And you only have 2 solar nerves. So this is not valid for repetitive tests. So for longitudinal assessments, how your nerve improves under treatment, this doesn't work, right? So this is just a once in a lifetime thing that can be done if everything else is not working. So we are not doing many cerebral nerve biopsies, but if patients had a cerebral nerve biopsy in the past, it is important to keep those specimens because they are so precious. So you can do a lot with it once you have it, but the indication needs to be really strict. Like you need to consider it before if it's worth it or not. So what is the consequence of a diagnosis or even an early diagnosis? So some hereditary neuropathies are treatable and others will be treatable in the past. So I'm at least a little optimistic for that. But examples for already treatable hereditary neuropathies are TTR amyloidosis or Fabry's disease. So there are diseases that are a step further along the line and we can learn from those. And since they are treatable, we need to identify them for sure. And then what I am really, what I think is really important to highlight here is that every hereditary neuropathy, whether genetically confirmed or not, deserves best supportive treatment. So to say that a neuropathy is not treatable at all is not true, right? We can do a lot, which is physiotherapy, occupational therapy, pain medications, orthoses. So if a doctor tells you, I can't do anything for you, this is probably not a good doctor, right? So there's a lot to do, even though there's no causative treatment. And then also knowledge. I mean, knowledge helps so much to make important choices. So if you know how your disease might progress, if you know how you will pass it on to your children, if you know that, for example, the probability of becoming wheelchair bound is quite low or is really high, you can make life decisions such as like choosing your profession or also getting counseling for your family planning. So knowledge is power. Knowing what you have is important for you, for patients. And of course, as I said earlier, it's the first step in order to develop new treatments, right? So from both sides, research and patient perspective, it's important to know what's the underlying cause. And then based on one's diagnosis, patients can decide to, for example, be enrolled in registries, and I mentioned some here, or trials, right? So this is also an important thing to consider. What if the genetic test is negative? I mean, that sucks for sure for all of us. But it doesn't mean that this is an unsolvable problem for the rest of your life. And I'm going to tell you a quick story here about a case that has a happy ending in the sense of diagnosis. So here in family 1, a female patient was enrolled in the Genesis database, which is also kind of a registry for genetic data. And she didn't have a genetic diagnosis. So she had a demyelinating CMT, but nobody knew the genetic cause. And there were many family members likewise affected with a dominant mode of inheritance. And 10 years later, a very nice and bright researcher, Danik Bayer, found this mutation in another 6 families. And suddenly everything made sense to us. We have not seen this variant before because it looks rather unremarkable. And the only way to find it and identify it as the genetic cause was to compare data sets to share them, to align them, and to see, oh, this is something that is repetitive, that is recurrent. So that seems to be something specific and important. And then we compared all those families clinically, and I helped with that. And then we noticed they actually all have the same type of CMT. And this is how this mutation and also this gene has been further elaborated. So this is important to know. So even if your genetic test is negative, don't give up. Share your data if you want to, of course. It's always your choice. And then there is a possibility that some researchers might find the genetic cause just a little later. When should family members or even children be tested? This is a really, really important question that I get asked a lot. One possibility is if they are affected themselves, then it's quite easy. It makes sense to test them too. Or if earliest recognition helps to stop or slow down the course and a disease-modifying drug is available. So that might even qualify for newborn screening in the future. So if there's something that can slow down the cause, it definitely makes sense to know as early as possible. Or if potential carriers just want it, right? If they express the wish to know. This is a bit difficult with minors. This is different in every country. In Germany, it's not allowed to test minors if they are not affected and there's no treatment. They can only decide for themselves if they are a little later. But then when they are later, they can still ask for it, like for the same reasons that we discussed earlier. What are the steps to get this this testing? Is genetic counseling is required. So patients need to be informed. And in some countries, there's even psychological support offered to patients or potential carriers before and after diagnostic testing. And I think this is really important to know, because the burden of a diagnosis can be quite demanding. Even if a test is negative, then you might feel something like survivor's guilt. So it's important to discuss this in your family before, and perhaps also take the chance to talk to a psychologist before. Doesn't mean that you have a mental disease, not at all, right? But it's useful to shed light on potential diagnosis before you actually know the result. And then information is key, right? So you need to be informed about the disease of interest, the molecular genetic testing method, so what test is going to be performed, and then potential outcomes. So some tests can either be positive or negative, but then there are many different tests that can also come back with an undecisive test result. So you don't even know. You might have some variants of unknown significance. This is a horrible word. It means that there is something, but clinicians and researchers for now don't know whether it is or not the cause of your disease. It could be, but they're not sure. So this is also something then individuals need to live with, right? To struggle with potentially this insecurity. And then also potential consequences of any of those results. For example, if you have a positive test result and you are not affected by the disease yet, but you know that you will get it at some point, what about insurance? Are you still suitable, right? Can insurance still take you? Are there treatment options? Yes or no? So this is also something that you need to think about before getting tested. And of course, there are counselors who can help you with those things specific to your disease. And then there's something I think also really important to mention, which is the right not to know. For now, until now, I think I only illustrated the perspective that everyone wants to know. And I think most people who come to webinars like this do want to know, but everyone also has the right not to know. You think like, okay, I have so much going on in my life right now, and this is just too much for me. You can always wait. You can always step back and say, okay, I don't want to know right now, maybe in 10 years, right? So this is your right. Everybody's right. And you don't need to feel pressured into a diagnosis by anyone. So we're coming back to this slide. This is kind of showing the whole loop of what we've been talking about. And at the end, I would like to show you one example that I think is quite exciting from the Miami lab. And this is Elizabeth Jacob, an MD PhD student who I worked with in my time in Miami. She's really smart and nice. And her project nicely illustrates how important it is, what I said here, to work together. So CMT4B3 is an extremely rare subtype of CMT that affects children. It's autosomal recessive, and it's almost a syndromal disease subtype that can also affect the eyes. Come with some MRI findings. So it's quite complex and it's not fully understood mechanistically. And in her project, in a very close collaboration with the CMT4B3 Research Foundation, Elizabeth and collaborators created the largest international CMT4B3 iPSC resource. iPSCs are stem cells, basically, out of which we can derive neurons in the lab, in the dish. And that means that we can actually test potential treatment options or understand mechanisms in a target tissue, which are motor nerves. So this shows that with careful coordination between multiple clinicians, scientists, and patients across the globe, it is possible to ship samples to one lab and to use very precious biological tissue for research. So this is quite an amazing process going on. And it is used for something very, very novel as well. A mini gene that is supposed to be designed first bioinformatically. You can see here the original structure of the MTMR5 protein that is encoded, that is responsible for the CMT4B3, and then a smaller cut down variant of this protein that could be created using a mini gene, like a smaller gene, because the original gene is too big to get it with a vector into cells. So this is a project that tries to supplement a missing protein by injecting a mini gene with the idea that the main functions of the protein will still be there. And this is right now being evaluated in cell culture in the lab, and an animal model is planned. And of course, at some point, we hope that this will be something that patients can profit from. For now, I think what is exciting is the concept of a mini gene to be tested in CMT. So this is very novel and cool, and could also be a potential pathway for other CMT subtypes. And then also illustrating that even in an ultra-rare disease, there can be some substantial research if people collaborate. Good, so we're coming to conclusions now. I personally think that despite all the bad news that there is not much treatment available right now, I think that this field offers exciting research topics. So I always hope and try to attract other researchers to this field, to spread the news that there's so much to be done. And one example is CMT SORT that illustrates how understanding of genetic causes and mechanisms helps to develop treatment options. So this is also really a good symbol to show that there's hope. Okay, molecular genetic testing is important to find the underlying cause, and that helps to discuss the available treatment options, but also to develop new treatments, and again, to participate in registries and trials, which is an important step to be ready for treatment. CMT is a clinical diagnosis. I said that earlier. I think this is really important to highlight. A negative genetic test does not exclude CMT. So if you have CMT or not is not a decision of a genetic test. The genetic test is supposed to find the underlying cause. And research networks are important to compare data sets and understand new patterns so that even in cases without a genetic diagnosis, there is a concept how future analysis might actually reveal the underlying cause. Treatment options are mostly supportive today, but that doesn't mean that they are not important, right? So it's still something that you need to be informed about and that clinicians need to be aware of. Future directions are based on collaboration. I said that a couple times today. Sorry for repeating myself. So between patients, researchers, clinicians, companies, and networks, it's a requirement to surmount obstacles, right? And obstacles can be genetically unsolved cases, but also lack of disease-modifying treatment. So this is my last slide, is a big thank you slide, again, highlighting all the many people who have supported me and have taught me and work with me and for me. And this is a nice picture of the Miami lab from the Miami PNS meeting. I could show so many other pictures, but I thought this is one that really shows a group of young, excited researchers. So to spread the news that there's more to come, there's hope for the next years, decades, that we can see some more. Okay, maybe I stop sharing at this point, so we can go to the discussion.
Yeah, please let me thank you on behalf of all the attendants. You cannot hear them. I'm pretty sure they're clapping their hands. Your presentation was really nice and clear. And thank you for stressing that collaboration is the key, because we are all rowing in the same direction. So we have to work together and go to the goal. So thank you. And thank you for your time as well. So we gathered some questions during the registration process. So I will ask you some of them. So there's a question about, is there something to prevent progression of the disease? Probably you already answered this, but if you want to ask something.
So the question was if there's something to prevent?
Slow the progression.
Yeah, yes and no. That depends on the exact genetic mechanism. And since there are so many different mechanisms, so many genes and proteins and cell types, pathways, tissues involved, there's not one answer for everything. Right now, there is no approved treatment for CMT on the market, but there are some trials going on that have the aim of modifying the disease course. So DMTs, disease-modifying treatments. And there's hope that, for example, for CMT sort, there will be a medication to slow down or stop the progression.
Okay, thank you. Another patient asked if endurance sport can harm endurance.
Endurance sport. Sports are good. We recommend to be active, actually, but in Germany, at least, we have this word that we say, there's a muscle pain threshold. So ideally, avoid having your muscles sore after exercise. So stop before the muscles get sore. And you can specifically train the muscles that are not affected yet. So, for example, thigh muscles or the trunk, in order to compensate for those that are affected, because then your overall strength will be better and you're going to be more stable. There will be less faults. There will be less complications. So exercise is good, and especially endurance exercise. But everyone is different. There's an individual muscle soreness boundary that should not be crossed.
Thank you, Maike. So we have hand raisers from Ingolf. Would you like to say a few words, Ingolf? You're allowed to use your camera and mic.
Yes. Do you hear me?
Yeah.
Yeah.
Oh, wonderful. So, Maike, I'm so happy to have you here, and thank you so much for all this revealing, interesting information. I've never heard this so comprehensive and so clear, and I think it was very, very helpful and valuable hour to listen to you. Thank you so much.
Thank you.
Let me just ask one question which came to my mind. If on the one hand your study reveals that the progress of the disease is either quick or slow, but continues very clearly one line, and you have shown us the picture.
Yeah.
And this is, of course, the result of many, many cases studied. What about then the influence and impact, real impact of intense sort of exercise or therapy, like physiotherapy and these kind of things? One of the patients may use this from a certain time onwards, very intensively. Others may not use it at all. So if the result is that the progress is always the same, so it would discourage me to continue my exercise and use my time for other business. Or is this wrong, this impression, because the statistics are just statistics and do not look at the individual cases?
Yeah, excellent question, Ingolf. I try to say the latter. It's definitely a big data set, and it's only from CMT1A patients, right? It's not applying to all CMT. It's only CMT1A. So it might be very different for other CMT subtypes because we don't have such data sets for other subtypes in that range. The thing is, I think that there are some patients who are very, like, as you said, very strict with physiotherapy, and they avoid complications, they avoid falls, they don't have fractures, they don't need longer immobilization. So they might have a slower course over time. And maybe they had parents that were really dedicated in bringing them to a physiotherapist from childhood on. I think that there will be some fluctuations for sure, but they kind of get diluted throughout the overall cohort statistically. So we try, of course, to get the purest subtype of natural history. So for this analysis, for example, we removed all the patients that had foot surgeries because we don't know by now, we don't know whether they impact the cause, if they actually help or if they worsen. Like it's not really, because also every foot surgery is different and every foot is different. So it's very difficult to standardize those results. So what we tried is to remove everyone who had confounders. Physiotherapy was not considered a confounder. Out of personal experience, I know that this is something that really helps patients. But I'm not, it like everyone tells me. So our outpatient clinic prescribes physiotherapy flat rate, like lifelong, always, to all of our CMT patients, because this is the thing that helps. So I think that many patients who have been seen in centers and been enrolled in a study like this actually had physiotherapy. So this might be a confounder. And then I also think that some of the effects that our subjective benefit are not being captured with this score that we have, right? Because we used this score as an outreach, but how stable you are, how you feel, the frequency of falls, this is something that is not being captured in this score. So I would definitely encourage everyone to continue with their physiotherapy. This is a great thing to do. And this is only like very broad idea of how the disease CMT1A in that case progresses over time, but very much zoomed out over 20,000 visits again.
Thank you. Thank you.
Sure, you're welcome.
If I can add something, with our national patient advocacy group and a group of researchers, we published something about the efficacy of rehabilitation. So yeah, we have started to gather some strong evidence and that this could lend a hand. So there is a question from the chat from Eric. He's asking what do you think about RTMS therapy? They use.
Yes, cool. I work on that right now for neuropathic pain. So RTMS is repetitive transcranial magnet stimulation. This is a method that is being applied to the brain. So you put a probe. Typically, it's the left side brain, but it can also be right side. And then you stimulate the brain, specific brain regions. For example, the motor cortex in a very specific pattern. So there can be like up to 3,000 stimuli in 15 minutes. But there are different protocols that we apply. And the idea is that it modulates the brain activity and also brain networks with a magnet impulse. So for a purely peripheral disease, which applies for most CMT cases, I think it's like, at least from my pathophysiological understanding, it's not a very promising idea because you stimulate the brain and not the nerves, the peripheral nerves. And you cannot cover myelination defects. But what we hope it can actually improve is how the brain how can I say it? How the brain focuses on specific problems or defects. And pain, for example, is something that is memorized. So the brain restructures and assigns more attention to the phenomenon of pain over time. So the longer you are in pain, the more intense the pain appears, even though the stimulus might not be more intense or might even disappear. So this is something that we try to modulate with RTMS, and we have a clinical trial on this for small fiber neuropathy in Aachen right now.
Great, thank you. Larissa, would you like to ask something to Dr. Dohrn? Hello.
Yeah, sure. Thank you so much. Yeah, thank you for the very informative presentation. It was great. And I just have a couple of questions. So the first one is, could you maybe elaborate on why nerve conduction velocity tests are usually not used as primary endpoints in clinical trials? Because from what I've noticed, usually such measures as ONLS or 6-meter walk tests are used, but somehow the nerve conduction velocity test is never used. And I've always been wondering why, because as you mentioned, it's a measure of how functional the nerve is, right? Like how well it works. So I just wonder why it is not used in clinical trials to assess the efficacy of the drug.
Yeah. That is a really good question. The problem with nerve conduction studies is that they only cover one section of one nerve. So they do not give information on the entire nerve and the entire nervous system. So some clinical outreach are more generalized, so they give a better impression of the overall status, functional status the patient is in. That is one thing. The other thing is that nerve conduction studies are really, really specific at diagnosis, but they are not as good. They are not correlating so much with the clinical course. So if the patient says, my disease got worse, I lost the ability to, I don't know, do buttons, or my foot drop got worse, this is not really covered very well with nerve conduction studies. And the reason might be that in a specific range, we can really see a lot, but at some point when the nerves are already quite degenerated, then there's not much difference that still can be measured, right? Because it's the peripheral nerves that are accessible with nerve conduction studies. We cannot measure proximal nerves, only distal nerves, right? And if they are already degenerated, the disease still goes on, but you cannot measure much there. This is one problem. And another problem is that's probably rather a historical thing, that there have been clinical trials, for example, on diabetic neuropathy, who did use nerve conduction studies as primary outcome parameters, and they failed. And probably the reason why they failed was that the outcome parameter that was defined as primary was just not a good choice. So for diabetic neuropathy, which is a sensory neuropathy that is distal in feet, if you choose to measure nerve conduction studies of the median nerve and you measure carpal tunnel syndrome, this is not a good outread. So those trials failed, probably because of a bad decision. So this is why I think now people are more like, yeah, restrictive and scared, but it's still usually an outcome parameter, but usually not the primary one.
Okay, makes sense. Thank you so much. And then one more question, if I may.
Sure.
So maybe you can tell me how long it takes before the newly CMT-associated gene is discovered and the point when it is included in the genetic panels, for example, because I've noticed that there are genetic panels, right? It's there are many genetic tests, and there is exome, sometimes genome if you want to go really crazy,
right.
And there are panels. So I just wonder how quickly this happens. Maybe it depends on the country and the regulations of the country, or whatever the internal processes are. But yeah, this is a very interesting kind of thing for me to understand. Let's say the SORD gene, it was discovered that it is associated with CMT in 2020, and then when did it actually get into the test so that the patients can actually be tested?
I really like this question. So what we try as a community is to work together with diagnostic labs. So for SORD, I remember that we sparked this information to some of the most important diagnostic labs before the paper was published. So I know at least because I collaborate with a lab that is located in Germany, CEGAT, they had it on their panels before the paper came out. Right, so that was really cool. That was SORD is a great example to show how it can work and be implemented fast. That's not always the case. It also depends on the gene and the method that is required to find the defect. If it's just in an exome, like something that can be covered easily, they maybe need to fine-tune their primers, but that's all. That's easy to be done. If it's a whole different new pattern mechanism, requires a completely different type of PCR that needs to be implemented, then it takes longer. And then there's also collaboration between the labs. So some labs say, we do this screening, but not that. So then the sample needs to be sent to another lab. So it's different, but we try, at least whenever we find new genes, we try to inform the labs and suggest to screen it. Sometimes for a certain amount of time, that was the case with SORD, scientific labs offer to do free of charge screening for scientific purposes for a defined time. Like for SORD, the Miami lab did that for a year or so. So that was free of charge, but that was not a certified diagnostic lab, right? This was just scientific screening. But to cover the time until the gene is on panels, and then patients can get retested to confirm the diagnosis in a diagnostic lab. That's ideal, right? For other genes, it might take longer.
Okay, got it. Thank you so much. Yeah, got it. Thank you.
Yeah, thank you, Maike. So there's a question from Henner. He is asking whether under certain condition, for instance, stress, lack of sleep, flu, COVID, there's a sudden acceleration in the worsening of the symptoms of CMT, as far as you know.
Yeah, there are definitely some environmental factors that can subjectively or even objectively worsen the course. The classical factors are chemotherapy. Like there are some medications that are really bad for the nerves. So we accompanied some of our patients through the process of cancer treatment. And that needs to be really fine-tuned with a neurologist and oncologist and all this. For COVID, we tried to assess that a little bit in our cohort. And at least we did not find that CMT patients are a high-risk category. At the beginning, we thought that. So at the beginning of the pandemic, we warned everyone not to come to the center in order to not contract the virus. But then later we observed by just sending out questionnaires to our CMT patients at our center that the frequency of contracting COVID was not higher, and that also the long-term outcomes were not significantly worse than in the general population. So I think this is worse for autoimmune diseases. If you have CIDP, that can be worse. I'm not sure, 100% sure about that, but I assume that autoimmune diseases are more prone to getting worse. But then, of course, if you become immobile, because you just lay in bed for a long time,
Exactly.
you lose muscle. You lose muscle. And for someone with a neuromuscular disease, whether it being CMT or muscle dystrophy or any myasthenia, losing muscle because of immobility is always a bad thing, and it's always more difficult to regain what you've lost. So from that perspective, I can totally see that it's not good to have any other chronic or long-term disease that has you bedridden.
Yeah, if you can choose not to have that, of course.
Exactly. Avoid it, ideally. I would recommend.
Sure.
But I had COVID too a couple years ago, and I was bedridden for like a week or 10 days, and I lost a lot of muscle. And
Really?
so it took a while to be back to normal, right? So independent of your comorbidities, it is not it's not easy. So try to avoid it, ideally.
Yeah, sure. So another question from Daniel is asking if you have any updates about the far next clinical trial.
The far next clinical trial has been stopped. So it was stopped late 2023. We were a center, so I know for sure, and it's been a big disappointment. Yes, so we were also center for the first for the Pleo trial, and then Premier. And the first one was not stopped, but many patients were removed, or many centers stopped because there were crystals in the solution.
Yep.
At some concentration. And then the second trial was stopped because it did not look like the outcome parameters would be reached. Very unfortunate. Yeah.
Statistical significance was lacking. We know that they were running a twin clinical trial in China, but we haven't heard from that so far. So, but as long as we will have news as a federation, we will keep you posted, of course. So there are 3 minutes more. If anyone has some other question, otherwise I'll have a couple. So just comment actually, you mentioned that CMT is slowly progressive. This means it's good news for the patient, but bad news for the researchers, because in clinical trials it's really hard to perceive the small changes that might take place during.
Exactly. Yeah.
During
and that means that, like, in reality, that means that we need many patients to participate in order to meet the statistical numbers that are required. First thing, and the second thing is we need longer observation intervals, like not just a year, longer, 2 years, 5 years. And that means, first of all, those trials are expensive, really expensive. That's a bummer. And second, it means that also patients need to wait a long, long time until we see results and can even fight for approval, right? So that's a disadvantage of this situation.
Thank you. Another comment is about the body mass index. We are recommended as CMT patients not to gain weight, but I was wondering whether the way that the body mass index is calculated for CMT patients is wrong, in the sense that we have a massive loss of muscles in our legs, so maybe our weight, our normal weight, is overestimated.
Yeah, I agree. I am not a great fan of the body mass index. I think in general, it's again, it's similar to Ingolf's question. If you look at the big picture, at huge populations, this is a really good outread for obesity, diabetes, all this. But if you look at an individual, it's not a good outread at all, because how we are built is so individual, like how much muscle you have or don't, right? So it's difficult to say. So I think it's much more important to move, to be active, to be like to have muscle, right? To have muscle. If not in your distal legs, then in your thighs, right? Or in your trunk, like to keep moving as much as you can. If you're in a wheelchair, still try to move whatever is movable, right? So and nobody is recommended to be obese. And if your skeletal system is already affected by a disease, of course, it needs to carry more, but also being underweight is not good, because then your body is burning your muscle. So
Sure.
starving situations should definitely be avoided. If you plan to lose weight, do it carefully, stepwise, implementing a lot of like endurance exercise if possible, right? Whatever is possible. Swimming is good because it's not so hard on the joints. So there are different activities, and you can also discuss this with the physiotherapist and go through the process together, right? Because it's an individual thing. You cannot generalize on this.
Yep. The very last question from Larissa, if it's quick, please, Larissa.
Yeah, thank you so much. So can you maybe think about why it has been more or less relatively easy to understand which genes are associated with demyelinating types as opposed to axonal types? Why is it so difficult for axonal types for us to understand which genes are causing axonal types? What do you think?
Oh, I think if I could fully answer this question, I would probably know how to find all these remaining genes,
Right.
right? Like what's the secret? I don't quite know. I think that still this is not supposed to sound arrogant, but I think that exons are still a little bit more complex than Schwann cells. Schwann cells are very specific for the peripheral nervous system, so I think it's easier to at least find out what they are doing and what they express. And axons are like really complicated, complex structures. Maybe they don't want to be understood by us right now, but it's a challenge. It's a challenge. We accept it, I would say.
Okay, thank you.
Well, so thank you to Dr. Dohrn for this nice morning and for your time, to all your attendants, to Ingolf. I didn't mention he is the vice president of the European CMT Federation, and of course to Applied Therapeutics and ND Pharma for their contribution to the awareness campaign. So follow us, and we're looking forward to meet all of you in our next webinars and events. Have a nice day and have a nice weekend.
Thank you. Thank you all. Have a nice day.
Bye.
Bye.
Goodbye.